Welcome to Biology My Passion. I am Soumya Harikrishna. We are studying the chapter Biological Classification, and also we dealt with the Kingdom Monera and Kingdom Protista. Kingdom Fungi divided into 4 classes: Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. Of this, we discussed the introductory part of fungus, the general features, their types of sexual reproduction and classification in the last video along with the Phycomycetes. Now we move on to Ascomycetes. The next fungi is Ascomycetes. Ascomycetes actually is the diverse group among the higher fungi. It has many members in it. It is actually 2 types. Either it can be thallus, which is non-mycelial, that means yeast. Usually these Ascomycetes are multicellular, but rarely it is unicellular. Which is that unicellular member? Yeast or Saccharomyces. So the yeast is unicellular, so it is amycelial, whereas the mycelial ones are Aspergillus, Penicillium, etc., which form tuft of hyphae forming mycelium. And we know what is the nature of the mycelium. It is branched and septate. Then asexual reproduction is through conidia. Conidia is a non-motile spore which is produced in a structure called a conidiophore, and it is exogenously produced. We discussed exogenously produced, endogenously produced, non-motile, motile types of spores in the previous video. If you haven't watched that, please go and watch that, learn the general characteristics first and then proceed further. Then yeast is the unicellular member. In that, asexual reproduction is by budding or a fission. Then their nature is saprophytic, means they are usually, as any other fungi, they are feeding on dead and decomposed matter. They can be parasitic, living on the body of other plants or animals, and they are coprophilous also. What is meant by coprophilous? Means the fungi or the organisms living in dung. Okay, so they are called a coprophilous. So these are the general characteristics of Ascomycetes. Now we will see the sexual reproduction, how it is happening. Before we move on to sexual reproduction, let's see how the conidia is forming. Suppose this is a hypha with a septum. From this, a stalk is coming up and it will produce structures like this. This structure is called a sterigma. From the sterigma, the spores are forming. What are spores? As we discussed, spores are thick-walled unicellular structures. Okay. Now this is called a conidiophore. Yesterday I told you sporangiophore is sporangia bearing stalk, right? The same way conidia bearing stalk is conidiophore. So the spore here will now detach from this and separate out. These spores are called a conidium. When the conidium falls on a substratum, if the conditions are favorable, it will start growing into a new hypha. Okay, this is how the asexual reproduction is happening. But if it is yeast, what happens? There is nucleus. First a protuberance will come out. This will go inside the protuberance, that is the nucleus extends. Then the nucleus will divide, that is karyokinesis takes place first. Division of nucleus is called karyokinesis. Then this will completely separate and now the bud will detach. So this is how budding takes place in yeast. Let's now discuss the sexual reproduction in Ascomycetes. So we learned that sexual reproduction has got 3 stages. First, a fusion of protoplasm, that is called a plasmogamy. That will result in 2 nucleated stage, that is dikaryon. This nuclei will fuse, then it is called a karyogamy, means fusion of nucleus. Since it is 2 nuclei are fusing, it becomes a diploid zygote. It will undergo reduction division to form again haploid spores. Haploid spores on germination will give rise to new hypha. Okay, so here we will see how it is happening. So this is one hypha. Okay, this is another hypha. They are of 2 different strains, plus and minus. Yesterday we learned they should be compatible for mating. Okay, then what happens? From this, one outgrowth is forming from one of this. So when outgrowth is forming, what happens? The nucleus will come inside. The same way here also the nucleus will come. So I will draw here little bigger. So this side is septum is produced. So the two gametes are forming. Now what happens? They fuse together to form a single structure. So I am not drawing the hypha now. I am just showing this inside. Okay. So now it is plasmogamy happened. So now two nuclei are there. This condition is called a dikaryon. Now from this, actually this will not fuse immediately. Now from this mitosis will happen, producing hyphae on both sides. But each hyphae having two nuclei. Right? The same way from here also. So like this many will come from here also. So ultimately it will look like a structure like this, a cup shaped structure. This structure is entirely made up of this hyphae. But what is the nature of the hyphae? All are having two nuclei or dikaryon. Okay. Now the penultimate one or the last one at the tip now will act as ascus. Okay. So here the name of the fruiting body is ascus. That is why this fungus is named Ascomycetes. Okay, so now the ascus is there. So there are a few ascus here. I will now draw ascus with a red. In between this other hyphae are there. They are sterile. So these are all ascus. Now I will show what is happening inside this ascus. Inside this ascus, sorry, here two nuclei are there. Okay, dikaryon. So now what happens? These two coming together inside the ascus. This ascus will fuse to form a diploid one. Okay. Then inside this diploid will undergo reduction division. So as a result, inside the ascus, how many spores will form? 4 nuclei will form. Each one is haploid. Okay, meiosis happens. As a result, 4 haploid nuclei are produced. Now, this will undergo mitosis. Mitosis will happen resulting in 8 nuclei. Each one is haploid. So these are, then each one will accumulate some cytoplasm and they form ascospores. So these ascospores will now be liberated by breaking of the ascus, and the ascospores on falling on a suitable substratum will grow into a new hypha and forming a new fungus. Here this structure from the male it is called antheridium, and the female this structure is called ascogonium. Okay. So the protuberance or the extension coming from the male or plus strain is called antheridium, minus strain is called ascogonium. Then this will fuse to form a single structure, but plasmogamy only occurs resulting in dikaryon. Then it will continue division to form a cup-shaped structure called called the ascocarp, which is the fruiting body. Ascocarp, carp means fruit, ascocarp, fruiting body, and at the tip the ascus bears dikaryon or the 2 nuclei. They are called ascus. Inside plural asci, ascus plural will be asci. Inside the asci the nuclei fuse, giving rise to a diploid nucleus, which will undergo reduction division to form again 4 haploid nuclei. That will undergo one mitosis. Here it is reduction division. Here what happens? Mitosis will happen so that 4 spores will become 8 spores. These 8 spores will be 8 nuclei. They will turn into spores. Since they are formed within the ascus, we call them as ascospores. Now they will be liberated out. The ascospores on falling on a suitable substratum will now grow into a new septate branched hypha. Now we have to see which are the main members of this group. First is yeast, which is unicellular member. Yeast is very useful in a baking and a brewing industry, making bread, wine, etc. Apart from that, there is a next organism called Aspergillus. Aspergillus can cause diseases, and also from this we commercially extract citric acid. At the same time, another one, Penicillium, the most common species, Penicillium notatum. We are all familiar with antibiotics. The first antibiotic, penicillin, was extracted from Penicillium notatum. Corals and truffles are the members which are edible, and they are considered delicacies. Another important thing to remember is claviceps and neurospora. Claviceps is called an ergot fungus. It can produce some chemicals which can cause hallucination. At the same time, neurospora is used in genetic and biochemical studies widely.